In terms of novel approaches, I think there are sort of probably 3 or 4 things that stood out to me. The first is that there's a lot of work going on in terms of developing the expression cassettes for various different gene therapies. So one of them is called the X5 for factor 8, which is to try and improve the expression ability of that particular cassette. So that's an interesting approach to developing and improving expression. There's also new developments in terms of new capsid developments along with expression. So Freeline are approaching that with something they're presented called FLT210. That's where the capsid's being modified and the expression cassette. So clearly what we're looking at is saying that gene therapy today is version 1. Maybe there could be a better, more designer version perhaps of the capsid and expression cassette to improve expression still further. The other 2 things that stand out are the development of a novel factor 9 variant that might be useful for management of haemophilia A with inhibitors via gene therapy. So potentially what we're looking at here is a factor 9 variant that can directly activate factor 10. So it gives an equivalent factor 8 activity. So that's an interesting concept that is going to take some thought, undoubtedly. And the last thing is encapsulated cellular systems that can be implanted that will produce factor 8. So these would be put in laparoscopically and have been used in mice and have now been shown to continuously express in mice for 6 months or so at quite useful therapeutic levels. So I think that's a really novel approach, putting in these small sort of capsules that can elaborate factor 8 on their own. So I think for me those are the 4 things: modification of the expression set, modification of the capsid, perhaps use of alternative variants that have additional features that could play into managing specific aspects of haemophilia, and then of course non-viral alternative systems like the extracellular systems, the little capsules. Clinical trials moving on from preclinical obviously takes some time. I think the one that's clearly going to be closest and we would look to see the soonest is probably going to be the encapsulated forms moving in.
I'm very excited about the approaches that have been taken so far with the AAV platform technology. We're in phase 3. We are getting close to product approval for at least one product. So the field has made dramatic progress over the course of the last few years. But I think we need to be thinking longer term than that. AAV is probably not the solution for curing hemophilia in the long run. We may come up with a better AAV vector system. That might happen, but more importantly, other technologies are starting to emerge as well. One technology is the use of lentiviral vectors, which would allow access to children and give us a permanent cure because the transgene, the factor 8 or the factor 9 gene itself, would be integrated into the chromosomes and would last for the duration of a patient's life. That's an important advance that is moving toward the clinic by several groups now. And then other approaches include non-viral gene therapy, the use of cell therapy to cure hemophilia by implanting cells that contain the factor 8 or factor 9 gene and having those cells churn out factor 8 or factor 9, just as they do in a bioreactor to make recombinant factor 8 or factor 9 protein. So there's a lot of excitement on the horizon from preclinical work. The preclinical research that's occurring with other platforms to cure hemophilia are moving forward toward clinical trials. I expect that lentiviral clinical trials will be initiated in the next 1 to 2 years. Those will be important because they will provide for long-lasting, durable, lifetime implantation of the factor 8 or factor 9 gene within a patient's cells to continue to make it for the rest of that patient's life. The cell therapy approach, where one has the factor VIII or factor IX gene in cells and then implants the cells, technology has been developed to make those cells stealth so that they can't be seen by the body and cause a huge inflammatory reaction, which is what would normally occur if we put foreign cells into our body. The cells themselves would be prepared to go into the peritoneal cavity. There's lots of room in the peritoneal cavity to put a lot of cells in that could make factor VIII protein. And the plan by the company that's pursuing this is to be in the clinic next year. So that's potentially very exciting as an alternative approach toward getting long-lasting factor VIII therapy to patients.